In continuation to Protozoa, the last phylum is Myxomycota. So in this Protozoa, in the previous lectures onwards, we have discussing four important phylum. The first one is Plasmodioporomycota, next one is Acrasiomycota, then Dictyosteliomycota, and the last one is Myxomycota. After this Plasmodioporomycota, this Myxomycota is one of the important and considerable phylum in Protozoa. Now we will see what are all the important characters and general features in Myxomycota. I am N.H. Shankar Reddy, PhD Plant Pathology. So Plasmodioporomycota already finished in the previous lectures, include Acrasiomycota. Instead of Chanjoa, we have discussed Dictyosteliomycota, and the last one is Myxomycota. This Myxomycota is generally known as true slimy molds. Now we will discuss the class Myxomycetes. Generally in Greek word myxa means slime, mycetes means fungi. That's why it's called as slime molds or true slime molds. These members of the fungi generally known as the true slimy molds or plasmodial slimy molds or acellular slimy molds. So these members of the fungi are generally considered as true slimy molds. So they contains almost 5 orders, 13 families, 62 genera, and around 800 species. Nearly 798 species are there. But we are going to see instead of 6 orders, because I added one extra order from other classification also for our further adding extra points and for further references for future as well. So generally, when it comes to this Myxomycota trophic phases, now we are going to discuss the common features. The trophic phase is actually a free living phase, which is actually a multinucleate, not single nucleate, multinucleate. Cenocytic saprobic plasmodium, which is shuttling kind of movement of plasmodium, can be observed. So this plasmodium, when sometimes may be converted into the sclerotium, maybe during unfavorable time or maybe during the required time, this plasmodium can be converted into sclerotium. Hope you guys know what is plasmodium. Oh, in the beginning of Plasmodioporomycota itself, we have discussed what is plasmodium, what are the different types of plasmodium. But still again, we will discuss what is plasmodium again the types also. Just to recall the things again, nothing else. Here the plasmodium means multinucleate mass of protoplasm is known as plasmodium. Here there are types of plasmodium that we will discuss in the later slides. Generally, these slimy molds are grown in dung. So if you see dung, this common general these slimy molds are used to grow, but this Myxomycota group of fungi plays a very important role in a food web or maintaining the food cycle in the ecology or in the environment. This Myxomycota group of fungi plays a very important role. And why these fungi are generally valuable? Because to study the organisms used to study the mitotic cycles or morphogenesis, chemical changes or the reproduction, structural physiology, movement of protoplasm. So for various kinds of biological purposes or various kinds of study related purposes, this Myxomycota group of fungi having a very great value. So before, as I told you, the types of plasmodium. So before entering into the types of plasmodium in Myxomycota, so whatever the types of plasmodium that we are going to see, the same kind of types of plasmodium we already discussed in Plasmodioporomycota also. For just recalling again, I just gave again the types along with a couple of extra information along with the diagrammatic representations also for easy understanding. As I told you, what is plasmodium? Multinucleate mass of protoplasm is known as plasmodium. Here I gave a little bit in a different way. It is a naked motile, it can locomote. Motile multinucleate mass of protoplasm that is enveloped by a plasma membrane, because it contains naked, it contains no cell or cell wall like things. It contains, instead of cell, it contains plasma membrane, and it is a gelatinous slime kind of sheath. If you see this, it is like a slime kind. If you just touch like a slime kind of things, are like a some kind of slime like sensation we can observe. That's why it's called as a gelatin slime like sheath. It covers with some kind of sheath, so that like that's why it is known as slime molds or like slime fungi, right? So generally this plasmodium means multinucleate mass of protoplasm is known as plasmodium. So this plasmodium, this gelatin slimy sheath feeds like amoeboid fashion. How amoeba will grow, whatever that direction of protoplasm or cytoplasm grows, the same way the amoeba also will start to grow, right? Start to move. So in the plasmodium, you just remember only one thing: multinucleate mass of protoplasm is known as plasmodium. There are three types of plasmodium in Plasmodioporomycota and as well as a Myxomycota or Myxomycetes also same. So protoplasmodium in Greek, protos means first. It is a first primitive type of plasmodium. Okay, aphanoplasmodium. Aphanis means invisible. It is an invisible kind of type of plasmodium. Phaneroplasmodium. Phaneros means visible. All are Greek terminologies only that we will discuss in detail about one by one. What is protoplasmodium? So here protos means first, right? As I told you, this is the most primitive type of plasmodium that is undifferentiated in a microscope. This is the microscopic picture. This is, you can see, most primitive type of plasmodium that is actually undifferentiated. You cannot differentiate this in a microscope. So undifferentiated mass that actually very smaller, less than 1 mm in diameter, and generally granular in structure. If you see, like a small granular structures can be seen, and having a slime kind of sheath. That's why it's called a slimy mold. So it having a slimy sheath. So generally this kind of protoplasm will give rise to sporangium. Imagine this is the in Mycota also, this that amoeboid or that amoeba gives rise to sporangium, right? Here in this protoplasm, there in acrasials, that is amoeba. Here in Myxomycota, see, if you see, almost all kinds of general features or properties are almost more or less similar, but the terminologies are differing, right? So there it is amoeba, here it is protoplasm, nothing else. Here this sometimes this protoplasm gives rise to sporangia, where the sporangium, so such kind of sporangial development or such kind of protoplasm where it can convert into sporangium can be observed in echinostelials and species like Lycia. So such kind of species where this sometimes maybe probably maybe this protoplasm gives rise to a sporangium. So this is about protoplasm. Let me repeat once again. It is a primitive type of plasmodium where it is undifferentiated in a microscope, generally covered by a slimy sheath. So sometimes this protoplasm gives rise to sporangium that we can observe in the members of echinostelials and some species of Lycia. The second one is aphanoplasmodium. As I told you, aphanis means invisible. So here it is at the initial stages, it is also resembles like a protoplasmodium only. In the later stages of development, there is a very clear set of branches development and the network of kinds of things can be observed, but it is a very fine, transparent and undifferentiated stance. Here if you see the picture, we cannot differentiate. It is also undifferentiated. And very in network of strands can be seen here, right? This protoplasm is actually non-granular when it comes to aphanoplasmodium. Sorry, when it comes to protoplasmodium, it is a granular. When it comes to aphanoplasmodium, it is a non-granular and a transparent. So its veins are generally like a slimy sheets. However, like protoplasmodium, it is also contains a slimy sheets. That's why it's called as slimy molds. All the members are like slime only. Okay, streaming is rapid, rhythmically reversible. So rhythmically reversible kind of streaming can be observed, and aphanoplasmodium is generally observed in the members of staminatales. So it is a characteristic of staminatales where it can produce aphanoplasmodia. Next one is phaneroplasmodium. These are all the last type is phanero means visible plasmodium. If you see this, it's like some kind of thick strands or granular cytoplasm can be seen, right? So generally it is an undifferentiated well advanced fan like threads that is actually exhibiting both ectopytic and endopytic plasmic regions are that is developed in the slimy sheath. So here the important point that we need to consider in this phaneroplasmodium is a shuttling kind of stream. Shuttling kind of stream is nothing but the cytoplasm actually grows for some kind of time. Imagine from this point onwards it is growing or it is moving from one direction. Imagine it is moved 9 centimeter in one direction, in the other direction also it will move in the same distance in the same period. So that's they are telling shuttling streaming means the movement of cytoplasm in one direction for some time, again it will go in the reverse direction in the same time they are mentioning time, not distance, just make it correct. So in the same time, if it is going one direction, the reverse direction also it will go in the same period. That is called shuttling streaming that can be observed in this phaneroplasmodium. So this type of plasmodium is most common in the members of Physareals. And other common features included if the environmental conditions are not suitable, this entire plasmodium starts to convert into pore. So that, as I already told you, when the unfavorable conditions occurs, this plasmodium starts to give like a sporangia or sporangiospore, where in acrasials this plasmodium gives, that amoeba gives sporophore, sauropore. There it is called sauropore, now it is called as sporophore. So which is again stalked. There it is called as, in acrasials also it is called stalk, here as also it is stalk. There it is called sauropore, here it is called sporophore. So under unfavorable conditions this plasmodium is converted into saurophore. But why this plasmodium is converting into saurophore, what is the exact reason behind this converting is still not known, still yet to be discovered. So the development of saurophore, this is, here is sporophore. In Myxomycota it is sporophore, in acrasials it is called saurophore. Okay, so in the development of sporophore is actually two types: subhypothalic and epihypothalic. Subhypothalic, hypo means under, beneath. Okay, so the development of a sporangium takes place beneath the hypothalamus. For your understanding only I have placed here. So development of sporangium starts from beneath. So the examples, members of echinostelials, trichials and physaries are the examples of this subhypothalic. When it comes to epihypothalic, this hypothallus is laid down actually on the surface of the substratum. You can observe here, like however actually they laid above the surface of the substratum. This substratum or plasmodium is there, they actually laid above the plasmodium. So where from the above the plasmodium the sporangiospore starts to develop it above the sporangiospore start to develop. The examples of staminales. So staminatales, see how this plasmodium is converting into sporangium. As just now I told you one point, under unfavorable condition this plasmodium starts to develop into a sporangium. So this is the plasmodium. Slowly this plasmodium is getting like little bit producing some primordia. How this primordia is producing? Due to the protoplasmic flow, whatever the direction of this protoplasm is going, this sporangia also start to develop. We can see there is some arrow mark kind of things we can see here, which means the protoplasm is moving in that direction, so that it will force it to produce this bulb or this kind of structure that is called sporangium. Here sporangium, again from sporangium the spores are produced. This is the phenomenon how this plasmodium is converted into sporangium. So this is the two types, subhypothalic and epihypothalic. Hope you guys understood how this plasmodium is generally converted into sporangium. So generally sporangium means it will start to produce spores, right? There are sporulating structures are there. This is all about so what are the types of sporulating structures this it will produce. So as I told you, under unfavorable conditions this plasmodium is actually converted into sporangiopore or sporangia. But the feeding plasmodium is actually converted into fruiting body, that is, the plasmodium is converted into fruiting bodies, or maybe the whole plasmodium is nutted. As I told you, why this plasmodium, at what time, what is the exact reason for converting this plasmodium into sporophore or sporangial development, of what exactly initiates or forces the plasmodium to get develop this sporangiospore is not known. But some factors like light, temperature, pH, moisture, extraction of food have been identified as one factor, but that is not the sole factor that is responsible for the conversion of plasmodium into sporocarp or the initiation or germination of sporangia. But still it is not known. There are some four important sporulating structures that we can observe in this Myxomycota. First one is sporangia, second one is plasmodiocarps, ethelia and pseudoethelia. Now we will see one by one. So the first one is sporangia. However, like in Mycota, sporangia can be seen, right? So here 1 to several thousand sporangia may be produced in a single plasmodium. So this is like a sporangia. The 1 to several thousand, hundreds of sporangia can be produced. So this sporangia is a globose, generally globose, cylindrical or cup-shaped cellophane, generally known as a hypothallus. So this stalk is there in the center. There is a small stalk is there. That stalk later extended to the sporangium. Along with the sporangium, that is known as columella. To understanding this, I have clearly given here. This is the stalk, right? Initial anchoring structure. in the later stages, this stalk only extends and it is anchoring the sporangium, right? So this extended stalk, or this stalk is generally an extended stalk along with this sporangium, is known as columella. For your understanding, I have given one microscopic picture also. This is the actual stalk. This stalk is actually extending along with the sporangia. This extended stalk along with the sporangia is known as columella. This is also an important feature in this sporangia. The second one is the plasmodiocarps. So it is actually, I think we have already seen in the types of plasmodium, aphanoplasmodium kind of thing, right? So this is similar to stalkless sporangia. There is no stalk, nothing can be seen. There is different kinds of branchings can be seen with the plasmodia. There is general habit. So hemitrichia is then a best example of this plasmodiocarp kind of sporangia. Next one is aethelium. Aethelium kind of sporulating structure. If you see here, it is actually cushion shaped fruiting bodies. All these cushion shaped fruiting bodies are fructifications are actually they try to make it as a fuse. Like we can also say some kind of fusing, but we can differentiate it. But generally, this aethelium consists of several sporangia. This is the individual sporangia. This is the individual sporangia. This is the individual sporangia. So this is a combination of this athelia is a combination of several kind of sporangia that tries to fuse together and during the evolutionary development, but we can differentiate it. So generally this athelia means several sporangia try to fuse into the one structure, but we can differentiate it during the evolutionary development. All the sporangia try to fix in a single together. So this is called athelia. The next one is a pseudoethylene. Pseudo means fast ethylene. Ethylene means all are combined to form a single kind of fruiting body. So the same kind of thing also we can observe here. Several sporangia are crowded. We can see here multiple number of sporangia are crowded together to start to behave like a sporophore. But even though the multiple number of sporangia are crowded together here, individual sporangia can be easily distinguishable. We can see like a bunch of grape bunch like things, right? So even though all the grapes are in a same bunch, we can differentiate the grapes, right? In the meantime, even though all the sporangia are try to crowded together like a same branch, we can differentiate this sporangia one by one. Generally, the sexual reproduction in Myxomycota is generally having two ways, homothallic and heterothallic way. Homothallic species example is Didymium squamulosum, and the heterothallic species is Physarum polycepharum and Didymium iridis. These are all the homothallic and heterothallic species of Myxomycetes. According to the Alexopoulos classification, there are 6 orders are present in according to Alexopoulos 1996. I added, not I, other scientists are added. That's why for further information, providing further information, I added one more order, so that becomes 7 orders totally. Alexopoulos gives only 6 orders, and further classification, Kirketer and the further classification, one more extra thing is added, so that is 7. So 6 plus 1, 7. Now we will see. Ceratomyxales are proteosteliales, lysiales, echinosteliales, trichiales, physareals, staminetales, protosteliales. These are all the different kinds of orders that comes under this Myxomycetes. We are not going to discuss in detail about all these orders. We are going to see just examples. So Ceratomyxales or proteosteliales example is Ceratomyxa, Lysiales, Lycia, Lycogala, Cribaria, Reticularia, and Echinostelium and Clastroderma, and trichiales, Archeria, Dynema, and Trichia favogeneus, Trichia scraba, Varia, Decipens, Botrytis, Flavoformis. These are all the various kinds of species that comes under trichials. And one important thing is the wasp net slimy molds. This Metatricha vasparum is generally considered as wasp net slimy molds. Sometimes we may expect exam such kind of questions. Then Physareals, most important thing is Physarum polycepharum. This is the most important fungi in biological studies. And Diderma, Badamia, Fulgo, generally called as blob, and Didymium. These are all the Didymium iridis. There are a couple of species that comes under this Physareals. Steminotis, and one more thing is prostelials, Clavostelium, Ceratomyxa, Echinostelium, or Prostelium. These are all a couple of examples comes under this Myxomycetes. These are all just examples only. So these are all the some pictures. Lycoglava, this is how Lycoglava will look like. This actually look like sporangia, right? So this is Comatrica, and Badamia, this is Badamia, and Dictyostelium, this is the Dictyostelium, right? So these are all the some important or couple of points that we need to consider in Myxomycota. This is Fulgoseptica, the picture of Fulgoseptica. Uh, so all the things that we have mentioned, this entire protozoa is like Plasmodium, Poromycota, Crassiomycota, Dictyosteliomycota, and Myxomycota. We mentioned the only important things. If we go in detail about each and every single thing, that will be very difficult. We are just concentrating on the exam importance along with theoretical importance, semester importance, and also with various kinds of competitive exams also. That's why we are giving just important points only, not in detail about. When it is required, definitely we go in detail. But in when it comes to protozoa, you guys need to remember the two things. One is Plasmodium poromycota, and the second one is Myxomycota. So these two things are the very important things. Put more I mean more specific concentration on Plasmodium poromycota, since that is the only thing that is in since years the question papers are maybe in exams also concentrating. Maybe in the rare cases we may expect questions from this Myxomycota only. If I want to say, this Acrasiomycota and Dictyosteliomycota are almost negligible, but I suggest you to have further knowledge, or to have gain the knowledge. I suggest you to go with every single point. And for further more information, student can refer my book, A Vision into Plant Pathology, complete student version. For further doubts and clarifications, students can reach us at www.geekyresearcher.com. Stay geeky and stay tuned. We are Team Geeky Researchers.